Let's start. Hi all. Thanks for the invitation. It's an honor for me to be here sharing with you. This talk is based on the study that I did for my undergraduate thesis, and the title is The Relevance of Spatial Scales and Biodiversity of Coral Assemblages. Okay. And this is particularly a case from Venezuelan coral reef. So the importance of scales in ecology have been largely acknowledged for decades. Ecological processes act on a variety of spatial and temporal scales and show up as a patterns. In other words, the issue of scale is critical for formulating general predictions about patterns in space and time. So in the past, manipulative studies were limited in time and space, but modern ecological thinking agrees that in order to understand the system, for example, the community, it is important to study it at the appropriate scale. So consideration of the scale is helping to address another relevant issue in ecology, the question of what influences the distribution and abundance of organisms. We all know that the distribution of the species depends on climate, species interaction, habitat structure, and dispersal ability, right? But it's pretty obvious that each of these processes acquires less or more relevance according to the spatial scale that we are studying. So generally, the presence or absence of organisms within a community may depend on rare or large-scale dispersal and colonization events, while local abundance is more a function of frequent or fine-scale processes such as biotic interaction and habitat heterogeneity. So this implies that the communities are structured by nested abiotic and biotic factors along different spatial scales that often occur along environmental gradients. So basically, if you know if there is a variation at different spatial scales and which one is the most relevant, then you can make inferences about what of those processes may be important to define what you are observing. And of course, these have an impact on efforts and management decisions, right? So let's be more specific. When we talk about the benthic community, these studies are very complicated. We know that marine habitats tend to be highly heterogeneous, and it's very difficult to identify natural units and establish the limits of these scales. So it's not an easy task. And studies have not been as extensive as in the case of terrestrial ecology. But what we know is that smaller spatial scale are the most variables in terms of diversity and abundance. However, there is a lack of consistency when the scale is increased, like the studies differ in which are the most important sources of variation after a scale. So being more specific, talking about the coral reef, we all know that coral reef represents the most complex and diverse communities in the sea. There is a compiling evidence indicating that ecological processes controlling the structure of coral assemblages, I don't know, for example, substrate availability or recruitment competition, are strongly depend on spatial scales. And in addition, oceanographic processes, which in partly define the environmental setting of a reef, are also extremely variable within habitat, across reef, systems, region, etc. So biological and environmental factors may interact with each other to produce different patterns in species distribution across several spatial scales. So in consequence, understanding the underlying factors controlling the coral species richness in a reef is not a simple task. Actually, it is a multi-scale problem. So let's go back to basic ecology. So we have the community structure that is composed by abundance and richness. And the total richness of a region is what we call gamma diversity. So gamma diversity can be partitioned into components. Alpha diversity, that basically is the number of species in a site. And then we have beta diversity, that is the variation of the species identity from one site to another. So for decades, ecologists has debated way to estimate and interpret alpha and beta diversity. But in recent years, the study of beta diversity has gained a lot of interest because of the question of what actually makes assemblages of species more or less similar to one another at different places and times. So scientists have developed many measures of beta diversity, but there is no consensus in which one are the most appropriate to answer particular ecological questions. This paper of Baselga in 2010, he partitioned the total beta diversity into components. Basically, we have nestedness and turnover. Nestedness means that you have this first side that has this pool of species, but then in the second side, we just find like a subset of this pool, and this is what he called nestedness. This side A1 and A2 are different for that. And then the other component is turnover. Turnover says that you have this B1 side that has this pool of species, and then in the second side, we found new species that we didn't see in the previous side. But the previous side, the side number one, have these species that we don't find them in the B2 site. As I said before, there are many measures of beta diversity, and Mary Anderson proposed that distance-based multivariate dispersion homogeneity tests can be interpreted as changes between sites. In other words, it is a beta diversity measure. So I prepared these schemes for show you guys what this means. So we have these points, and each point represents a pool of species from each site. And then we have the centroid, that is the expected value of the pool of species for each location. So we found that are points that are really close to the centroid and another that are far away from the centroid. So when the points are close to the centroid, this means that this site share more species with the expected pool. And if the point is far away from the centroid, this means that this site shared less species with the expected. So we can have two cases. The case when we have all these points really close to the centroid, the distance is basically the same, and this is when we have less multivariate dispersion. And we can have the case that we have a lot of multivariate dispersion, and we have points that are really close, others that are really far away from the centroid. And in terms of beta diversity, basically this means that if we have less dispersion, then we have low beta diversity. And if we have more dispersion, then we have higher beta diversity. Because in the first case, this case, well, all the sites have more or less same species. And in this case, we have a different pool of species in each site. So in coral communities, there are few studies that focus on beta diversity. And Venezuela is no different. Issues about spatial scales or changes in the species composition have been little explored using hierarchical designs. So Venezuela is a good place to do this study. It is a natural laboratory to study these changes in the community structure. We have 4,000 kilometers of coastline, actually the largest in the Caribbean, and it's a highly heterogeneous coast. We have 12 points of upwelling that represent like a longitudinal gradient. So our main question was, is the benthic community, and in particular the coral assemblages, are variable at different spatial scales? And if the answer is yes, then which are the most relevant scales to explain this variability. And does the coral species composition change at this spatial scale? And maybe there are patterns in this species change. So the study area was along the coast of Venezuela. We divide the coast in 3 regions: west, center, and east. And we have these localities in each region. And it's interesting because we have coastal reef, we have continental island, and oceanic island. So the experimental design was fully nested. We have this first random factor that is called region. Within each region, we have 2 or 3 localities as a random factor 2. Then within locality, we have 5 sites, and we deployed 4 transects in each site. So the fieldwork was carried out between 2017 and 2018. The depth was between 5 and 12 meters, and we took 60 photos per site. We used the GCRMN protocol, but we did some modifications. And what we did was deploy these transects of 30 meters parallel to the coast, and we took pictures every other meter. So for each site, we have 4 transects, and in each transect, we have 15 photos. Then we went to the lab, and we analyzed these photos that we took in the field using the PhotoQuad software. So the routine was basically measure this reference, then we draw the quadrant, and we deployed 25 random points, and we have to identify the organisms under these points. So in this form, we obtain the absolute abundance of large groups, absolute abundance of corals, and present absence of coral species. We use Primer and RStudio for the data analysis. In the case of spatial variability, we use a Breit-Curtis index, a PermaNova to test variability at different spatial scales, and we did an analysis of principal coordinates to see the patterns. In the case of beta diversity, we use the coral present absent data. We use Jacquard similarity coefficient, the PERM-DIS, that is the test that I explained before, to measure the changes in species composition, and we use the R package beta part to the partition of the Jacquard index. So we took a lot of photos like this one that have this huge acropora in the middle of the picture. So we were worried that maybe we were underestimating rare species, right? So we did the sampling coverage test to see what was going on. So sampling coverage is basically the relationship between interpolation and extrapolation of the effective number of species. And this allows us to make statistical inferences without the bias of underestimate rare species. So good news, we found that 80% of the sites has sample coverage greater than 0.7, so it is a proper sampling effort. We don't have to worry about these huge corals. So let's jump into the results. This is a general description of the great groups that we found in the coral reef of Venezuela. I'm so sorry, I'm noticing that the labels are in Spanish, but I will translate it for you guys. So this is absolute coverage, and each point represents an observation in a quadrant. So it's pretty obvious that algae are really abundant in coral reef from Venezuela. And when I say algae, I mean like turf algae or cyanobacteria. Then we have the abiotic component, that is basically sand, rocks, bare substrate, etc. Then we have the scleractinian corals, octocorals, and fleshy algae. When we talk about scleractinian corals, we did this plot with relative coverage. We found 19 genera and a total richness of 28 species. And again, each point represents an observation in a quadrant. So we see that Colpophilia natans, Orbicella faviolata, and Pseudodiploris strigosa are really important in the Venezuelan reef. And then Madrastis mirabilis, Montastrea cavernosa, Porites astroides, and Siderastra siderea. So these are the results of the permanova. We obtained the greatest variability at the scale of site, which accounted for 35% of the total variance. Region was the second most important source of variation in the analysis. So these results indicate that coral assemblages in Venezuela only vary by 21% at the scale of region. Also, we found statistical significance at the scale of location within region. So our results indicate that coral assemblages are much variable at small to medium scales rather than hundreds of kilometers along. This is a principal coordinate ordination plot. Over 70% of the total variance was explained by the first three PCO axes. And basically what we found here is that center, that is purple one, and the east are really similar to each other when we compare with the western region, that is the yellow one. The simpler analysis show clear patterns defining regions and location. However, sites show variable species composition and cover. Overall, western region was largely composed by Orbicella faviolata, whereas the eastern reef were dominated by Pseudodiploria strigosa. On the other hand, we have the central region, and it's mixed coral communities. Remember that in the central region, we have like this oceanic reef with coastal reefs. So these species accounted for more than 70% of the dissimilarities across localities, sites, and regions. And it's really interesting, this result that we obtained, because the effect of upwelling and other oceanographic processes has been pinpointed as a strong factor shaping the assemblage of coral communities here in Venezuela. But our studies show that coral assemblages are in Venezuela are much variable within and between localities that we originally expected. So as I mentioned before, modern ecological thinkings agree that the issue of scale is really important, but in the Caribbean, we don't have many papers studying this using hierarchical designs. Basically, all the papers use an univariate perspective to this issue. So we found these papers from Venezuela are not about coral reef. Actually, this is of epibionts in mangrove roots. They found that higher variation for these assemblages were found at the smallest and biggest scales. And this other paper is actually in rocky shores in Venezuela, and they found that these communities are highly variable at tens of kilometers and not between localities or regions. So this paper highlights the importance of local processes in providing structure to different assemblages of sessile organisms in Venezuela. It is widely acknowledged that behind patterns are ecological processes that shapes communities. In this case, our patterns are that all spatial scales are variable, and site is the scale that explains the greatest variability. So the processes that shape these patterns can be different, like at the small scales, processes such as predation and competition have really important impact on species abundance and composition. For example, the abundance of urchins or the selection of the specific selection of species on the reef fishes. And of course, issues like structural complexity may influence competition and survival of colonies depending on the sizes. And this have an impact, for example, in settlement of at medium and large scales, then the processes may be changing. Like we know that in Venezuela, anthropogenic stressors are really relevant. So this can alter biological interaction, thereby affecting the structure of coral assemblages. And of course, environmental stressors such as sedimentation, upwelling, etc. So now let's talk about biodiversity. When we assessed biodiversity, we found that the highest variation in species present absence occurring between transects of the same site and between sites of the same locality. Oh my gosh, okay. On the other hand, at large scale, we found no significant dispersion in species composition between localities of the same region. We found basically the same when we did the dissimilarity index, Jacquard index. So site and locality were the spatial scale with the largest Jacquard dissimilarity. That is this circle in purple, with the turnover component as the main contributor. That is this yellow point. So furthermore, at large scales, Jacquard dissimilarity decreased, and the contribution of turnover and nestedness became evener. So the results indicate that coral species found between the western, central, and eastern region of Venezuela can result either from a species replacement or from a species loss, which is interpreted as a subset of the total pool of species, right? So we know that beta diversity is scale dependent, but most of the studies assume that it's homogeneous at the small spatial scale. So in our study, we found that more likely to have different species composition within transects of a single site than between localities belonging to different regions. However, it is not clear what are the conditions favoring this heterogeneity within the reef sites. This variation at the smallest scale could mean that Venezuela coral assemblages are in good condition, as well as there are patches of mortality with more or fewer species reflected in a high turnover rate. And at large spatial scales, we found quite similar coral species composition, which maybe in part can be due to the reduced pool of species that exist in the Caribbean when compared to in the Pacific region, for example. So in conclusion, coral assemblages structure in Venezuela is highly variable at different spatial scales, and particularly a small spatial scale seems to be very important. But we don't know what is causing these patterns, right? So there's a lot of work that has to be done, much remains to be explored. So the processes that could underlie these patterns are diverse, are complex. And when we talk about beta diversity, coral assemblages are fairly homogeneous across the Venezuelan coast, but when we increase the spatial resolution shows greater heterogeneity. And the species turnover seems to be a very important component of the diversity and is a relevant phenomenon to explain this diversity patterns. So in other words, this study showed that the structure of coral communities in Venezuela is probably regulated by a series of interconnected processes that act individually or jointly at different spatial scales. So that's all. I want to take this time to say thank you to the experimental ecology laboratory members that helped me along this journey and helped me to carry out this study.